in a worrying sense, you're just a temporary collection of carbon atoms that was once atmosphere, but now they're you, whatever that means. Molecular archaeology, and that's looking at trying to find molecules like DNA and proteins and things like that that survive in the archaeological record. So, and using those to answer archaeological questions. I did my PhD, my training in bone diagenesis, so that was looking at how bones fall apart from a chemical point of view, how they sort of fossilize. Why do they survive in some places? Why they don't survive in other places? Why, for example, here in Atapuerca, why do you have such exceptional preservation in the caves there and those kinds of things? So we're trying to understand that more. And what I started doing there was looking at DNA preservation and collagen preservation in bones and trying to figure out whether we could try and predict those things a bit better. Because if we can, at the time, this was very early in the ancient DNA world, and we didn't want people to go around grinding up precious bones and teeth without any real chance of them getting any positive results. So if we could predict where we would find DNA, we'd say, okay, go to these sites and do those ones, but don't go to these other sites and do those ones. Now I do stable isotope work, so I analyze mainly bone, collagen, and sometimes mummy hair, and to try and figure out, broadly speaking, what people have been eating. So sort of what they call paleodietary research. If you don't investigate it properly, you just assume that all fossil bones are the same, and they're not. That was one thing that we discovered. So bones can be very well preserved, and it doesn't necessarily depend on how old they are, but very much the environment that they're in. How long they're exposed on the surface, the particular environmental conditions, the soil conditions. One of the things that we were looking at for the bone and DNA preservation was temperature. And we tried to predict those kinds of things using thermodynamics, basically. In terms of the preservation here in the Sierra de Atapuerca, for example, you've got nice deep caves, and caves are really good. They buffer temperature as well, and they're very stable. They're also nice deep sediments there, and all these factors help buffer the temperature. So they're very, very stable environments, and that might have something to do with the excellent preservation as well. I mean, the principle behind the stable isotope work about investigating paleodietes is you are what you eat. So the carbon and nitrogen in your body that makes up your tissues, that comes from, well, the carbon, for example, comes from the carbon dioxide that goes into a plant. You eat a plant, or you eat an animal that's eaten that plant. Those very same carbon atoms then become part of you. Okay, so in a worrying sense, you're just a temporary collection of carbon atoms that was once atmosphere, but now they're you, whatever that means. There are different types of environments, different types of plants, and the marine environment is slightly different. So we can try to track, if you eat a lot of marine food, you'll have a particular carbon isotope signal. If you eat a lot of if you're in a temperate environment, you'll have a different type of carbon signal. If you're in a warmer environment, you'll have a different type of carbon signal. So we can start to detect that in tissues. So in archaeology, we're looking at that bone collagen again that can survive for 50, 100,000 years. And we can analyze this, and we can figure out what people have been eating. Now, what I've been doing is I've been developing a technique that's based on something called liquid chromatography isotope ratio mass spectrometry, which is a very long-winded term, but the usual technique is to use solid samples, okay? So we take a chunk of bone collagen, that could be like a milligram. Doesn't sound like a lot, but it's still a reasonable amount of sample. Whereas with the liquid chromatography isotope ratio mass spectrometry, we can take a much smaller sample. We can use like 40 to 50 micrograms. So we're trying to reduce that damage that we do to the bone. So we could take a much smaller sample, basically. Okay, so we're applying the LCRMS technique to some of the material here from La Sierra de Atapuerca on this Mesolithic Neolithic transition. There seems to be very much a tradition of hunting, gathering, and having a lot of marine protein or freshwater fish as well. So that seems to be the way. Inland they might have more terrestrial diets, but certainly in coastal sites they got all these things. As soon as the Neolithic comes in, there's this transition and everybody has terrestrial diets. I guess that matches up very well with Christina's work on the genetics, that it's not that people are deciding not to eat fish anymore, they're just different people. These are people who've come in with farming and that's what they do. That's their economic system. Because there's no reason, fishing is easy, relatively speaking. Farming is hard work. So why would you abandon that way of subsistence? And the reason is you're not abandoning it because you're different people. In the Chalcolithic and the Neolithic, they're all isotopically very, very tight. It's very boring. Everyone's doing the same thing for about 4,000 years, and they're not eating fish. I'm trying to look for other types of samples. So we've been developing some work looking at stalagmites. So again, there can be tiny amounts of organic carbon that get trapped when stalagmites form. The other thing that really excites me, my PhD student Alicia has just finished working on some material like this, is we can analyze hair. And your hair grows about 1 centimeter a month, and we've analyzed some mummy hairs. And the thing with the bone collagen is it's a very broad look at your diet over several years. With a hair, you can sequentially analyze, so we can do this forensic analysis, and then we can see their sort of weekly, or fortnightly, monthly changes in their diet. So that's been really interesting. You can see seasonal patterns in people's hairs, beautiful changes across the seasons.